For decades, human history appeared to begin at a clear, reassuring point. Around 200,000 years ago in Africa, the first true Homo sapiens emerged. Before that moment were other human species. After it came us, a clean boundary, a recognizable beginning. But ancient DNA has erased that boundary. From a fragment of bone no larger than a matchbox, scientists recovered genetic material dating back nearly 200,000 years. What that DNA revealed was not a simple origin story, but a population already shaped by deep splits, long separations, and forgotten encounters. By the time anatomically modern humans appear clearly in the fossil record, their genetic history was already old. This discovery did not announce itself with fanfare. There was no single headline declaring the end of a theory. Instead, it arrived quietly embedded in data that did not behave as expected. Lineages diverged too early. Genetic distances were too large. Populations that should have been closely related were not something had happened long before the moment we once called the beginning. The implication was unsettling in its simplicity. If humans already carried deep genetic structure 200,000 years ago, then our story did not start there. It stretched further back into periods where fossils are rare. Landscapes have changed and entire populations may have risen and vanished without leaving visible traces. Ancient DNA does something archaeology alone cannot. It exposes absences. It reveals migrations that left no tools behind, separations that left no skulls to measure, and bottlenecks that erased almost everyone who lived through them. In this case, it showed that early Homo sapiens were not a single small group waiting to spread, but a mosaic of populations, sometimes isolated, sometimes reconnecting, across vast regions of Africa. This realization forces a shift in perspective. The question is no longer when modern humans appeared, but how many times our ancestors nearly disappeared before that appearance became visible. The discovery does not rewrite human history with spectacle. It reshapes it by showing how fragile, fragile, and contingent our origins truly were. To understand why this changes so much, we need to look at the version of human history that existed before DNA was able to speak at all. Before ancient DNA entered the picture, the story of early human history was built almost entirely from what survived physically in the ground. Fossils provided shapes and proportions. Stone tools hinted at behavior. Sediments and animal remains helped reconstruct environments. From these fragments, researchers assembled a framework that felt stable, even if incomplete in this framework. Modern humans emerged in Africa roughly 200,000 years ago. Fossils, from sites in eastern and southern Africa, showed skulls with rounded brain cases, reduced brow ridges, and faces that looked recognizably human. These remains were placed at the beginning of our species, marking a transition from earlier forms of Homo to Homo sapiens. The assumption was that once this transition occurred, a relatively small population carried those traits forward. Africa itself was often treated as a single evolutionary stage. While regional variation was acknowledged, it was generally assumed that early modern humans formed one loosely connected population. Differences in skull shape or tool style were interpreted as local adaptations rather than evidence of deep separation. Over time, this population expanded, diversified, and eventually left Africa in waves that peopled the rest of the world. Encounters with other human groups were understood but seen as limited. Neanderthals in Europe and Western Asia, and Denisovans further east, were viewed as evolutionary cousins who diverged earlier and followed their own paths. When genetic studies of living people later revealed traces of Neanderthal and Denisovan ancestry, those findings were framed as brief episodes of interbreeding at the edges of expanding modern human populations within Africa. However, the prevailing assumption was continuity that was lasting during the emergence of Homo sapiens. As a result, the continent's deep interior history was often described as poorly resolved rather than fundamentally complex. Missing data was treated as absence, not as a sign of unseen diversity. Timoline reflected this simplicity. Population splits were placed relatively late. Bottlenecks, when discussed, were usually associated with migrations out of Africa or with environmental crises. Tens of thousands of years later, the earliest phase of Homo sapiens was imagined as a foundation period stable enough to support what came next. This view was reinforced by the limits of earlier methods. Fossils are rare, and preservation is uneven. Entire regions leave little trace of human occupation, especially in tropical environments where bone degrades quickly. Stone tools, while abundant, are often difficult to link directly to specific populations or biological groups. Without a way to track ancestry directly, Researchers relied on patterns that could be observed, measured, and compared. Even when questions arose, they tended to stay within these constraints. Why did some early fossils look more archaic than others? Why did tool traditions vary so widely across Africa at similar times? These puzzles were acknowledged, but rarely seen as evidence of deep population structure. The dominant model still favored gradual change within a broadly shared lineage. By the early 2000s, this narrative was deeply embedded. Modern humans emerged in Africa around 200,000 years ago. They were relatively few in number, but cohesive. Over time, they expanded, encountered other humans, and became the sole surviving branch of the human family. What this model could not account for was information that never fossilized. It could not see populations that left no bones behind, migrations that ended without descendants, or separations that lasted tens of thousands of years before reconnecting. It assumed that the visible record, though incomplete, was broadly representative. Ancient DNA would challenge that assumption directly. It would show that absence in the fossil record did not mean absence in history, and it would reveal that by the time Homo sapiens appeared as a recognizable form, their genetic past was already far more complicated than the traditional picture allowed. The breakthrough came not from a dramatic new fossil, but from a change in what scientists could extract from old bones. For most of the 20th century, DNA was considered too fragile to survive deep time. Heat, moisture, bacteria, and radiation break it down quickly. In many environments, especially in Africa, organic material decays rapidly. The idea of recovering genetic material from remains tens or hundreds of thousands of years old seemed unrealistic. That assumption turned out to be wrong. Advances in molecular biology made it possible to isolate tiny fragments of DNA preserved inside dense parts of bone, particularly the inner ear region of the skull and the roots of teeth. These areas can act as natural vaults protecting genetic material long after the rest of the skeleton has degraded, instead of long intact strands. Researchers learned to work with fragments sometimes only a few dozen base pairs in length. The process was meticulous. Bones were cleaned to remove surface contamination, often ground into fine powder, and treated in sterile laboratories designed to prevent modern DNA from entering the sample. Chemical solutions released ancient DNA fragments, which were then copied millions of times using specialized enzymes. Computer algorithms reassembled these fragments by aligning them to reference gums from living humans and other ancient populations. Crucially, scientists also learned how to recognize authentic ancient DNA. Over time, genetic material accumulates characteristic damage, especially at its ends. These damage patterns act like dawns, allowing researchers to distinguish genuine ancient sequences from modern contamination. What once seemed impossible became routine. As ancient DNA data grew, they began to extend deeper into the past. Neanderthal genomes were sequenced from remains over 40,000 years old. Denisovans emerged from a single fingerbone found in Siberia. Each discovery reshaped the human family tree. Then came African ancient DNA. Recovering genetic material from Africa proved far more difficult due to climate and preservation conditions. For years, the oldest African genomes were only a few thousand years old, offering limited insight into early Homo sapiens. But gradual improvements pushed that boundary back. Genomes from individuals tens of thousands of years old revealed population structures that had not been visible before. The most jolt results did not come from a single complete genome, but from patterns shared across many partial ones. By comparing ancient African DNA with gummies from living populations, researchers could infer population splits far older than the samples themselves. These methods act like genetic archaeology, reconstructing events long before the earliest preserved remains. One of these inferred events pointed to a major separation within Homo sapiens. Around 200,000 years ago, possibly earlier, genetic lineages diverged far deeper than expected, if all modern humans descended from a single recent population. Some lineages showed signs of long isolation lasting tens of thousands of years before reconnecting. This was not a minor adjustment to the timeline. It suggested that early Homo sapiens are already divided into distinct populations across Africa, with limited gene flow between them. These groups experienced different environments, different precedents. Others shrank or disappeared entirely. In some cases, genetic signals hinted at admixture with deeply divergent human groups whose fossils have not yet been identified. These were not Neanderthals or Denisovans, but African populations that split from the Homo sapiens lineage much earlier and later rejoined it. Their existence had been invisible to archaeology alone. Dating these events required careful calibration, right combined mutation rates, radiometric dating of fossils, and comparisons with well-dated ancient Yum outside Africa. While exact years vary between studies, the overall pattern is consistent. By around 200,000 years ago, modern humans were not emerging from a single cradle, but from a landscape already shaped by deep genetic history. What made this discovery so powerful was its independence from physical appearance. Fossils can look similar even when populations are genetically distinct. DNA revealed relationships that skulls could not. It showed that anatomical modernity did not mark the beginning of our species story, but rather a visible phase of a much older process. This realization set the stage for a fundamental reassessment. If early Homo sapiens were already genetically diverse, already shaped by isolation and reconnection, then many assumptions about migration, extinction, and survival had to be revisited. The DNA was no longer just filling gaps. It was revealing a hidden structure beneath the entire early human timeline. What emerged from these genetic reconstructions was not a simple branching tree, but a braided history of separation, survival, and reconnection. Ancient DNA revealed that early Homo sapiens did not evolve as a single population steadily refining modern traits. Instead, they existed as multiple groups spread across Africa, often isolated from one another for long periods of time. These populations were not marginal variations within a unified whole. Genetic distances between some of them were as large as those seen between modern humans and Neanderthals tens of thousands of years later. This meant that by 200,000 years ago, human populations had already been evolving along partially independent paths. Some groups lived in relatively stable environments, maintaining continuity over long spans of time. Others faced repeated environmental stress. Africa during this period was not uniform. Climate shifted dramatically, transforming grasslands into deserts and back again. Rainfall patterns changed, lakes expanded and vanished. These changes fragmented populations, cutting off contact between groups for generations at a time. DNA carries the signature of these separations. Long periods of isolation reduced genetic diversity, while sudden reconnection leave distinct signals of mixing in the ancient Yums. Both patterns appeared repeatedly. This told researchers that early humans experienced cycles of fragmentation and reunion, driven by environmental change rather than linear expansion. One of the most striking findings was evidence of severe population bottlenecks. At several points in early Homo sapiens history, effective population sizes dropped to remarkably low levels. Some bottlenecks appear to have lasted thousands of years, during which only a small fraction of a population contributed genetically to future generations. Such bottlenecks explain a long-standing mystery: why modern humans, as a species, show surprisingly low genetic diversity compared to many other mammals. The answer is not that our species is young, but that our ancestors passed through repeated filters of survival. Entire populations disappeared, leaving no direct descendants. What survived was a narrow genetic sample of a once broader human landscape. Ancient DNA also revealed unexpected patterns of interbreeding within Africa itself. For decades, interbreeding was framed as something that happened after modern humans left Africa when they encountered Neanderthals and Denisovans. Genetic evidence now shows that admixture was already part of our history long before those migrations. Some early Homo sapiens populations mixed with deeply divergent human groups that split from the main lineage. These groups are sometimes described as ghost populations, not because they were mysterious, but because their physical remains have not yet been identified with certainty. The genetic traces, however, remain embedded in living people today, especially in certain African populations. This finding challenged the idea that Homo sapiens replaced all other humans cleanly within Africa. Instead, it suggests a process of absorption and blending, where traits and genes from different human groups contributed to what we now recognize as modern humanity. Evolution was not a single stream, but a network migration. Patterns also had to be reconsidered, rather than a single expansion out of a core region. Early humans appear to have moved back and forth across the continent repeatedly. Some groups expanded into new areas only to retreat later. Others remained localized for long periods. Genetic data showed that geographic distance did not always predict genetic difference, indicating complex movement patterns. Crucially, these findings altered how researchers interpret fossils. A skull that appears anatomically modern does not necessarily represent the ancestor of all later humans. It may belong to a population that contributed little or nothing to future lineages. Conversely, populations that left few or no identifiable fossils may have played a central genetic role. Early African fossils often display a mix of modern and archaic features. Rather than representing transitional stages in a single lineage, they may reflect different populations at different points along parallel paths. Anatomy alone could not capture this complexity. The timeline of human origins became less about a single moment and more about a long interval. Anatomical modernity emerged gradually across different regions, influenced by local conditions and genetic exchange. By the time fully modern traits became widespread, the underlying genetic foundations had already been shaped by deep history. Perhaps most importantly, ancient DNA demonstrated that extinction was a dominant force in human evolution. Their disappearance was not necessarily due to inferiority or replacement, but to chance, climate, and demographic vulnerability. Survival often depended on small advantages compounded over time. What we call modern humans are the descendants of those populations that happened to endure through multiple crises. It was contingent, fragile, and shaped by events that left little visible trace. By revealing this hidden structure, ancient DNA did not simply adjust dates or add detail. It overturned the assumption that our species emerged as a unified group. Instead, it showed that humanity is the product of many histories, most of which ended before they could be seen. This deeper understanding reframes everything that comes after migration out of Africa, encounters with other human species, and the spread of culture all rest on a foundation far more complex than previously imagined. And that complexity carries consequences for how we understand ourselves. Today, they change how we understand what it means to be human. If early Homo sapiens were not a single population but a network of groups, then modern humanity is not the product of one origin, but of survival across many fragile beginnings. Ancient DNA shows that our shared ancestry does not come from uniformity. It comes from repeated mixing after long separation. Populations that evolved apart for tens of thousands of years eventually reconnected, exchanging genes, behaviors, and adaptations. Traits that seem universal today are the result of this convergence, not of a single ancestral blueprint. This helps explain why modern humans, despite low overall genetic diversity, are remarkably adaptable. Our ancestors were shaped in different environments, under different pressures. Long before global expansion, when these lineages merged, they carried with them solutions to survival that had already been tested in isolation. The result was a species prepared by chance rather than designed for a wide range of conditions. These findings also dissolve rigid boundaries between ancient human groups. The distinction between Homo sapiens and other humans becomes less about clear replacement and more about degrees of connection. Within Africa, early modern humans did not simply outcompete others, they absorbed them. Genetic traces from deeply divergent populations persist today, meaning extinction was often partial rather than absolute. Survival was not a straight line from primitive to advanced. It involved repeated losses. Entire populations with their own cultures, technologies, and genetic identities vanished without descendants. What remains is not a record of superiority, but of contingency. Ancient DNA also reframes discussions of human variation. Differences among modern populations are shallow compared to the deep structure that once existed within our species. Groups that today appear closely related are separated by far less genetic distance than early Homo sapiens populations were from one another. This emphasizes how recent our current diversity truly is. Perhaps most importantly, these discoveries highlight how connected all living humans. The deep divisions of the past did not persist. They were bridged. Every living person today carries ancestry shaped by multiple ancient populations that once lived apart. The idea of a single pure lineage has no biological foundation. The 200,000-year-old genetic signals uncovered by ancient DNA do not tell us who we are in moral or philosophical terms. They do something more grounded. They show how survival shaped our species through vulnerability rather than dominance, through isolation rather than unity, through reconnection rather than replacement. In this light, humanity's origin is not a moment of arrival, but a long process of endurance. The people who lived at the dawn of our species were already heirs to a deep and complicated past. We are heirs to theirs. What remains is to reflect on how this knowledge reshapes the broader story of human history, and why ancient DNA has become one of the most important tools for understanding where we come from. For much of the past century, the origins of modern humans were framed as a beginning, a point in time where something new appeared and then moved forward. Ancient DNA has quietly shifted that frame. It shows that what we once called the beginning was already the result of a long and unstable history. 200,000 years ago, early Homo sapiens were not standing at the start of a journey. They were standing in the middle of one. Behind them lay periods of isolation, population collapse, and survival against changing climates. Around them existed other human groups, some closely related, others deeply divergent, whose genetic traces would eventually blend into what we now call humanity. Ancient DNA matters because it records what fossils often cannot. It preserves relationships rather than appearances. It reveals absences as clearly as presences. In doing so, it reminds us that much of human history unfolded without leaving clear physical evidence behind. Entire populations lived, adapted, and disappeared, shaping our ancestry without shaping museum displays. Instead, it places them within a larger framework. Bones show us who lived where. DNA shows us who endured, who mixed, and who vanished. Together, they tell a story that is less tidy, but far more accurate. What emerges from this deeper view is not a dramatic revision driven by speculation, but a careful recalibration based on evidence. Human history did not simplify as it moved forward. It narrowed through repeated bottlenecks and chance survival. A small subset of ancient diversity carried the future. Modern humans are the result of that narrowing, not the product of a single successful population, but the descendants of many histories compressed into one surviving lineage. Ancient data does not change who we are. It changes how clearly we can see the path that led here.